Okay, this is where we left off from Chapter 9, Part 1. So this is Part 2 now. So next we'll talk about the individual microscopic anatomy of a skeletal muscle fiber. So if you look here, we talk about first within, we talked about the myofibrils. So within the myofibrils, there are going to be smaller structures known as filaments. So these are going to be within, of course, those myofibrils. There's smaller structures, of course, within the myofibrils. They're going to be contain thin and thick filaments. So these thin and thick filaments, of course, they overlap each other to an extent, which creates these striations. If you look down here on the picture, you have the thick filament and your thin filament here and here. It is going to be, of course, involved in the contractile processes. It's going to be arranged in compartments called sarcomeres. And sarcomere is going to be the area from one Z disc to the other Z disc. So this area here is going to be a sarcomere. So sarcomeres are the basic functional unit of a myofibril. So make sure that you know that for your test. The basic functional unit of a myofibril is a sarcomere. Next we just talked about the Z disc to Z disc. That is the area or region that separates one sarcomere from the next. The M line, of course, is in the middle here. So that is going to be in the middle of the sarcomere. It's supporting proteins that hold the thick filaments together. And just to kind of give you an idea of the sizes of the thick and the thin filaments, the thick filament is about 16 nanometers, whereas the thin is about 8 nanometers. So the thick filaments will be about twice as thick as the thin filament. But this picture here is just represents, it shows the relationship of the zones of that muscle fiber, this filament. So next we have your checkpoint questions, blank. And always, when we get to the checkpoint questions, just kind of try to answer them first on your own or pause the video and answer them yourself. And then if you can't answer them, then just let it play and I'll give you the answers. But number one, the myoglobin, of course, binds oxygen molecules that diffuse into the muscle fibers from interstitial fluid. Number two, your answer would be the Z disc, separates one sarcomere from the next. Your answer for number three would be myofibril is a contractile organelle of the skeletal muscle. So going back to more details of your filaments, you have your contractile proteins, which are your myosin and your actin. Both of these are going to generate force during your muscle contraction, so that's their function. Okay. First, we'll talk about myosin. Myosin, of course, is the protein that makes up the thick filament, so make sure that you know that myosin is thick filament. Your actin is going to be thin, which I'll give you on the next slide. Protein that makes up the thick filament. If you look up here, we see an individual picture of one. So it does contain one tail, and if you notice here, two heads. Okay, so it binds to myosin binding sites on the actin molecules of a thin filament during your muscle contraction. So it is a thick film. Thick filaments in general contain about 300 myosin molecules. So they are a lot more. These are just seeing one individual one here. So what they did is they took this thick filament here and enlarged it, and just to show you one individual one. And they contain myosin. So actin, of course, is your protein that is a main component of the thin filament. You have it's on each actin molecule is a myosin binding site where myosin head of a thick filament, of course, binds during your muscle contraction. So there are three different thin filaments. Sorry about that. That does not look very good. I'll try to rewrite that. It's kind of hard to write on a computer screen, so I apologize for my handwriting. So you have three different types of your thin filament, or thin filament is going to contain three. And again, one is going to be your actin. We've already talked about that. And actin, of course, is a contractile protein. And then I'll put number, 2 and 3 up here. 2 and 3, of course, you have your troponin and your tropomyosin. And both of these are going to be regulating proteins. Okay, and then make sure that you know that both of these are going to be regulating proteins. Okay, so we'll talk about the regulating proteins. Your troponin and your tropomyosin, these are going to be what they refer to as regulatory proteins. That is a component of the thin filament. What that means is these proteins are going to help switch the muscle contraction process on and off. One will turn it on, the other one will turn it off. So the first one here we're going to talk about is your tropomyosin. And this is when the muscle is relaxed. It covers the myosin binding sites on the actin molecule, thereby preventing the myosin from binding to the actin. So it's going to turn it off. The troponin, of course, is when the calcium ion binds to the troponin. It undergoes a change in shape. So this change moves tropomyosin away from the myosin binding sites of the actin molecule, and a muscle contraction is going to begin. So it's going to turn the muscle contraction on, whereas tropomyosin turns it off. So they're opposing actions. So it's going to begin as the myosin binds, of course, to the actin. This picture here is showing you a representation of what they refer to as your sliding filament mechanism of your skeletal muscle contraction. So I'll kind of make sure that you understand how this works, or the process, or the pathway. Okay, and you can put it in any words that you like. But first, just to let you know, of course, the first little picture here is showing you, of course, that the muscle is relaxed. There's no contact between the actin and the myosin. So that's what's happening first. Okay, then what happens is your myosin head is going to attach to the thin filaments. Is going to be, and then when it does that, it attaches. It's called cross bridge. Okay, so then your second picture here, you're seeing that the myosin head has come across, the thin filament has come across and attached. Okay. So once it attaches, of course, to that thin filament, what happens is this walks along, and as it walks along, it makes the contraction. As it walks along, it shortens it. So I always ask, students on the test, does the thin and thick filaments change shape? It does not actually change shape. What they do is they change size according to the walking or the cross bridges. So the cross bridges form, and as those actin filaments move closer together, it'll shorten the muscle, but it does not change its shape. Okay, so it's still the same shape, and then of course when it relaxes, it'll go back. So of course what happens when like I said, the cross bridges, it walks along the thin filaments at both ends of a sarcomere, pulling that filament towards the midline. Okay. So just remember and write this down some words. Your thick and your thin filaments do not change shape, and the key word there is change. Okay. But what it does is the two discs, of course, are closer, of course, and the sarcomere shortens. So it does shorten, but it does not change the shape of it, other than shortening it. So here is a summary down here of this process. So the skeletal muscles shorten during the contraction because they thicken and thin filaments slide past one another. And I'll post this one. I do like to show, if you're in class student, I would show a video of this in a visual in process or in motion. So I will actually, and I think I posted some lab videos for you that show this as well too with a model. But I'll also I'll try to find some animations and post those online for you as well too, just so you can see this in action. So does the thin and the thick filaments change? They don't actually change shape, they just shorten. Okay, so they don't change, but they do shorten or lengthen. So no, they do not change in the size. And that's because, of course, the sarcomere is shortened. So sarcomere shortens and causes the shortening of the whole muscle fiber, which in turn leads to shortening of the entire muscle. Next, we have your contraction cycle. So you have the onset of the contraction begins with the releasing of your calcium ions. This is how it begins, releasing of those calcium ions into the muscle cell. So the calcium ions allow those cross bridges that we just talked about to form between the actin and the myosin. Okay, so where they bind to actin, opening the myosin binding sites. And again, I'll post the videos up as well as the contraction cycle as well too. So first picture here show you what they refer to. Make sure that you know this process. I like to kind of give little shorthands of each one. So the first one, I would say my shorthand of this one would be ATP hydrolysis. Because I always tell students when you have to learn a pathway and the details of it, if you have one key, memorize the keywords in order and then memorize what goes with the keywords. So sometimes that can help you to memorize the whole process a little bit easier. So ATP hydrolysis is number one. So what happens here? This is just what we just went over with that one model. So the myosin heads hydrolyze ATP and become reoriented and energized. Okay. So the second picture is part of what we just went over as well too. And so we're going to call this one number two, formation of the cross bridges. And I'm just going to abbreviate that because I'm running out of room. But what happens is the myosin heads, of course, are going to bind to the actin, forming the cross bridges. So what it happens is, remember, it pulls that actin filaments together, and it causes the sarcomere to shorten. Third one here, we're going to call it power stroke. Okay, for some reason my pen quit writing, so I apologize. So the third one, just write power stroke. That is when their myosin cross bridges rotate towards the center of the sarcomere, called power stroke. So the sliding, and that's just the sliding of the filaments. Number four, of course, this one we call it just detachment of the myosin from the actin. So you can just call it detachment. And again, my pen is not writing, so I apologize. Detachment, of course, of the myosin from the actin. So as the myosin heads bind to the ATP, the cross bridges, of course, are going to detach and form the actin. So what happens here is this is a process that repeats over and over. So the contraction is going to repeat, or the cycle is going to continue if the ATP is available and the calcium ion levels in the sarcoplasm, of course, is high. So the length and the tension relationship indicates how the forcefulness, of course, of a muscle contraction depends on the length of the sarcomeres within a muscle before contraction begins. When a muscle fiber is stretched, there is less overlap between the thick and the thin filaments, and the tension, which is forcefulness, is going to be decreased or diminished. So your muscle fiber, of course, can be stretched to 170% of its optimal length. There is no overlap, of course, between the two filaments when that happens, so you cannot contract. So the zone of the overlap and each sarcomere is optimal, and what happens is the muscle fiber cannot develop its maximum tension when that happens, so it diminishes when there is no overlap at all. So this will be the end of part 2, and I'll start part 3 at the neuromuscular junction.